So I really want to thank the foundation for giving me the chance, not only by receiving the award, but also by being here and telling you about this really work in progress that started only a few months ago. I'm very excited about the data I'm going to tell you about. So my lab is a stem cell lab. Let me see. I am the co-director and founder of the Center for Regenerative Medicine, so we do a lot of stem cell work. The whole facility has been built in the second floor of this building to work with human stem cells. So we really try to do at a very large scale this production of this very particular type of stem cells that were mentioned before, these pluripotent stem cells. And it's really a discovery that changed the way that we can think about how we can study now diseases. It was a discovery made in 2006 by Dr. Shinya Yamanaka, who got the Nobel Prize in 2012 for this discovery. My lab has been working for many years now in how to create these iPS cells, or induced pluripotent stem cells. And really, I wouldn't be here today if it wasn't for this person, Alice Anani. So I happened to meet Alice a couple of years ago in Israel. She's the founder and president of the CDG Foundation in Israel. And she basically really convinced me, as she's been trying to do for many years, to get into this field and to start doing research in CJD. And I was very lucky to have a neighbor in Boston University, Dr. David Harris. Probably many of you know, he is a previous recipient of this award. And Dr. David Harris has been doing a lot of work on the biochemistry and understanding PRP proteins. So the project that we decided to work on has three main components. The first one was, can we establish a library of induced pluripotent stem cells from the familial individuals, some of them containing the mutation, the E200K mutation. And if we can do it, can we now take these pluripotent stem cells and can make neurons out of these cells? And one particular characteristic of these pluripotent stem cells is that we know that they can go into any direction, form any cell type in our bodies, including neurons. If we can do it, then maybe we can use these cells to characterize the E200K PRP mutant protein in these neurons. And we can check how actually the protein behave in the neurons, how it traffic, and also if it's infectious. We can also do genetic and transcriptional studies. That means we can study the whole sequence of the cells, and we can also study how the genes are expressed in these cells. And maybe if we can find differences between the mutant and the non-mutant, we can study the mechanism of the disease, which is something that we're still trying to understand. I think it's very obvious from all these talks that there is a lot to be learned about the mechanism by how PRP actually creates neural damage. And if we can do it, then maybe we can also use this platform to study the toxicity in the neurons and hopefully maybe start testing new compounds. Some of them were already previously described by Dr. Harris using mouse models. So let me tell you a little bit about the project itself, what we have so far done. We chose the E200K mutation mostly because of Alice. But the truth is that this is the most common frequent form of the familiar form of CJD. It actually happens in more than 70% of the cases of familial CJD. It was originally described in the early 90s, and it's a change of lysine to glutamine in the codon 200. And the highest incidence of this type of CJD happens in a group of Lebanese Jews, mostly living in Israel, and with an incidence that's about 100 times higher than in the general population. I actually traveled to Israel a couple of times, and I brought with me many samples of peripheral blood. My laboratory at BU was one of the first to actually show that we can only use few mLs of peripheral blood to make these pluripotent stem cells. So I basically brought with me, frozen in dry ice, many samples from individuals, some of them carriers of the mutation, some of them not, but all of them pertaining to this big family. And there are a couple of interesting samples from these individuals, number 29 and 40. They are 96 and 95 years old, and they have the mutation, but they never contracted the disease. We call them super resilient, and we believe that maybe by studying their sequence, we also be able to study sequences that can be protective. So I took all these blood samples back to the lab, and we started doing reprogramming. And that's how we call the process of taking blood or skin and make them become pluripotent stem cells. And we actually made some nice progress. We already made iPS cells from 7 of these samples, several clones from many of these individuals, and we are also, as I'm speaking now, we're currently reprogramming many more. Some of them are non-carriers, so we believe it's going to be a good control to have members from the same family that they don't have the mutation, and we can do the control between the mutants and the non-mutant cells containing a very similar genetic background. So that was the first part. The second is, can we make neurons? And there are many different ways that you can do it. We focus on one of the original protocols developed in Cambridge, in England, by the Leipzig lab. And it's a very robust protocol, meaning in most cases it works well. And this is not trivial in the field of iPS cells. It's a very long protocol, and it involves many, many different steps with many different factors that you need to put in vitro in the system. But the cells actually differentiate very nicely. So I'm going to show you a few pictures of how we know that the cells are becoming what we call cortical neurons, and there are different types of cortical neurons. But the cells first become what we call neural progenitors, and these cells tend to grow, forming these structures called neural rosettes that you can see here in the pictures. And you can do some staining from some markers that are typical of neural progenitors, including nesting in green here, and also a transcription factor called PAC6 that's supposed to be in the nucleus. So what you can see here is our nuclei of these cells, and they're stained for nesting and PAC6, very beautifully forming these neural rosettes. An important aspect of this protocol is that this stage of neural progenitor, you can freeze the cells down and put them in the freezer, and then it saves actually 30 to 40 days of the protocol, because you don't need to go back to the pluripotent stem cell. You can now use the progenitor every time you need to do the experiments. And we've been doing this successfully too. But then you take the cells to follow up on the differentiation, and they start expressing more and more specific markers of neurons. By day 65, this is what we call a young neuron. So you can see a positive expression of, for instance, VGLUT1. And VGLUT1 is a transporter that is present in the synaptic vesicles in the neurons. But some of the more mature markers, like SATB2, is only very, very low expression. We can start trying to test the function of these neurons, and this is a classic assay called patch clamp, where you can actually run a current through the neurons in vitro, and you test if the neuron reacts to it, and that's called an action potential. And we were very happy to see that we can actually detect action potential, but this is not a normal action potential. This is what we expect if the neurons were young, as it is the case here. And now we took the cells farther down to day 95, and now you can see much more robust expression of the more mature markers, including this SATB2 and MAP2, and we are in the process of repeating now the patch clamp with these more mature neurons, hopefully in many of these different iPS-derived neurons. So that was a very reinvigorating that we can actually validate the neural differentiation. This is one example of now we're taking a normal, and this is a non-mutant line, compared to 4 CJD samples, all of them becoming neurons. This is day 95 and express very robustly all the markers. What about PRP? So these are type of questions that actually were very difficult to answer before in human cells. If you have an example of a neuron that you know they have both alleles, most of these samples are heterozygous. That means that they have one gene that is normal PRP, PNRP, and one mutant one. And we don't really know exactly what happens when these two genes are being expressed and producing the protein, one the normal one, one is the mutant one. So the first thing we wanted to know are our neurons expressing the protein, the PRP protein. And this is done with an antibody that detects both the normal and the mutant form. And yes, the answer is actually the cells express very robustly PRP. This is a typical staining of PRP in neurons. And you can see all of these samples expressing very nicely PRP. We also confirmed this by Western blot. But more important are the mutant cells expressing also the PRP scrappy, which is, of course, critical for our model. If we want to see that we want to study the disease in vitro, we need to see expression of the scrappy form of it. And indeed, there are a few ways you can do it. I think there was previous mentioning of some of these. So this is a positive control only. These are a cell line called N2A, and it's a neural cell line that you can grow in vitro. And you have the version that doesn't express the mutant form, and this is the version that expresses the mutant form, the SC scrappy N2A. And what you do a staining for PRP, and that's in red. And in addition, you add a thioflavin S, which has a high affinity to bind only to the PRP scrappy. And you can do it before and after PK treatment, that it was explained before. And the PK treatment will get rid of most of the normal PRP. So you can see all the red protein disappearing after PK treatment. But importantly, in the mutant scrappy one, you can see the presence of this yellow, very bright yellow accumulates of PRP scrappy. So when we did this in our neurons derived from the CJD, different from the ones from the normal one, we can start seeing very nicely accumulation of these yellow dots representing precipitates of the scrappy protein in our neurons. So we were able to differentiate the cells. We were able now to prove that they are expressing PRP and also PRP scrappy in the case of the mutant cells. So what can we do now? So I'm going to show you a few examples of what we've been now starting to do to see if we can detect abnormalities. So one thing that you do, you can do a staining with a substance called phalloidin. Phalloidin binds to one protein expressed in the cytoskeleton of all these neurons, and you can study the morphology of the neurons, one particular part of the neuron called the dendrite, and you can see if you can find differences. So there are many, many studies showing that part of this neural degeneration is associated with changes in the dendrite morphology, and one of the most important parts of these dendrite morphology changes is in the dendritic spines. And what are dendritic spines? So the dendritic spine, I put here the definition, is a small membranous protrusion. It's basically a small bump that you can see in the dendrites of the neurons. And there are many studies, including from David Harris' lab in mouse models, that show that there is a degeneration of these dendritic spines in the neurons of overexpressing PRP scrappy. So we only have started now to analyze our cells for this presence of these dendritic spines, and we're very happy to actually see that we can detect abnormalities, but we need to do more studies, we need to do more numbers and more clones. This is only the beginning, but we're very already optimistic about what we're finding in the studies we did. And lastly, we really hope that, so these are the conclusions of what we have been presenting so far. We were able to successfully reprogram many IPS lines from this E200K group of patients. We were successfully differentiated these IPSs into cortical neurons in a very robust manner. It takes 100 to 120 days, so it's a very long-term experiment to keep in culture, and a lot of things can go wrong, but luckily I've been working with very talented people in the lab, and the protocol is working really well. We were able to show robust expression of PRP and also PRP scrappy in the neurons. So now we're currently trying to characterize these cells in a very consistent manner to see if we can detect morphological changes and molecular changes. We are studying a collaboration with Regenerative, who offer for free to do whole exome sequencing of all our samples, so we will be able to also study the genetic sequence of these cells, comparing mutant and non-mutant, and also probably these two super resilient samples. And finally, we hope that if we can detect the differences, maybe we can start testing some of the compounds that were previously described to influence the conversion of PRPC into scrappy and also prevent some of the neural toxicity effects. So the most important slide, the acknowledgement. So everything that I showed today was really a collaborative effort between Sunny Park in my lab and a very talented postdoc, Nat Lee, in David's lab. They worked together to create the library and to make the differentiation happening. And I mentioned already Alice, who really was instrumental in getting me into this field. And I wouldn't be here today if it wasn't for you, the families, the donors. I really want to thank each one of you, the Gary Butterman, the Chuck Fear, the Cheryl Molloy, the Jeffrey Smith, and the Sassy Curry Memorial Grant for your contribution. Thank you very much.